Welcome. The altered metabolism or the cancer cell metabolism. One of the key ways cancer cell differ from the normal cell is its metabolism. The altered metabolism in the cancer, how it allow cell to obtain energy from altered sources. This is called altered metabolism. The reprogramming energy metabolism has been identified as an emerging characteristic in cancer cells. For cancer cells to sustain uncontrolled proliferation, they must make adjustment to their energy production. What's metabolism? Metabolism, as we know from biochemistry, the sum of the chemical reactions that take place within each cell of the living organism and that provide energy for vital processes and for synthesizing new organic material. Living organisms are unique in that they can extract energy from their environment and use it to carry out activities such as movement, growth, development, and reproduction. But how do living organisms do their cells extract energy from their environment, and how do cells use this energy to synthesize and assemble the component from which the cells are made? This is the metabolism, and there is anabolic processes and catabolic processes. This is full detailed explained in the biochemistry, but what we need to focus on is the energy metabolism, or the difference between the normal cell and the cancer cell in its assembly of the glucose. The normal cell, in the normal cell, metabolism is geared toward maintaining homeostasis, growth, tissue specific functions. Nutrient availability and quality play a crucial role in regulating normal cell metabolism. Cells extract energy from their environment, use it for activities such as movement, growth, reproduction. This process is enzyme mediated. Cell convert nutrient into molecule required for growth and maintenance. These reactions involve the oxidation of carbon atom from nutrient, generating energy in the form of ATP, adenosine triphosphate. ATP serves as the primary energy currency in the cells. This occurs in the Krebs cycle. On reverse, cancer cells exhibit distinct metabolic adaptations. Their metabolism is geared toward biomass protection and proliferation. Cancer cells consume more glucose to generate energy and the materials supporting cell divisions. The Warburg effect describes how cancer cells convert most glucose to lactate even in the presence of oxygen. This diversion of the glucose metabolites from energy production accelerates cell proliferation but generates only 2 ATP molecules per glucose. Metabolic adaptation in cancer cells. Cancer cells adapt to nutrient at the depleted environment by employing various metabolic strategies. The nature of these adaptations depend on the cell origin, transforming lesions, and the tissue context. Select metabolite not only serve as substrate for energy and biomass generation, but also regulate gene expression and influence neighboring non-transformed cells. Tumor can affect and be affected by nutrient distribution within the body. The cancer cell rewire their metabolism to promote growth, survival, proliferation, and long-term maintenance. The common feature of this altered metabolism increased glucose uptake and the fermentation of the glucose to lactate. This phenomenon is observed even in the presence of completely functioning mitochondria, and together is known as the Warburg effect. Glucose converted to pyruvate and lactate Cancer cells do this by finding and using alternative sources and alternate metabolic pathway. Normal cells typically break down glucose in process called glycolysis and convert it to pyruvate, which provide adenosine triphosphate or ATP for the cell. This is largely done in the mitochondria. However, cancer cells are very different, where they can convert glucose to lactate irrespective of the oxygen. Molecular cancer research also reveal cancer cell metabolism is a fascinating area of research that shed light on how cancer cells generate the energy they need to grow and spread. The Warburg effect. The Warburg effect refers to the observation. This is the name of the researcher or the scientist who discovered this phenomena, Otto Warburg, in 1930s. Unlike normal cells, use glycolysis, the breakdown of glucose in the presence of oxygen. Otto Warburg in a period noticed this phenomenon in the 1930s when he found that cancer tissue slices produced large amount of lactate from glucose even when oxygen was available. While some cancer cells do respire through tricarboxylic acid, the Warburg effect remains a hallmark of many cancers. These criteria, metabolic pre-programming, cancer cells undergo metabolic reprogramming to meet their increased bioenergetic and biosynthetic demands. They alter their flux through various metabolic pathways, ensuring survival and proliferation. Key metabolic alterations include decreased glycolysis, altered mitochondrial function, and a change in nutrient utilization. LDH glycolytic enzyme plays a crucial role in the Warburg effect. It's a transcriptional target of the oncogene MYC and is necessary for increased glycolysis and tumorogenic potential. LDHA inhibition has been explored as potential therapeutic strategy. Tumor microenvironment, nutrient availability within the tumor microenvironment influence cancer cell metabolism. Cancer cells adapt by reduce nutrient scavenging mechanism to sustain proliferation. Other cell types with the tumor microenvironment, endothelial cell, fibroblast, immune cell, also impact tumor progression. A number of activating mutation and enzymes that help exacerbate this process allow cancer cells to divert metabolite for useful anabolic processes such as mitosis. There is a look at cancer cell metabolism next to the normal cell metabolism. Here the pathway of the normal cell, and this is the pathway of cancer cells, and then this Otto Warburg. One of the first scientists to make this observation was Otto Warburg. This is known as Warburg effect, where normal cell takes in glucose, break it down by glycolysis to pyruvate, and mitochondrial undergoes the Krebs cycle, which produce 36 ATP molecule and CO2, where cancer cells undergo aerobic glycolysis, produce low ATP and lactate, which is important for cell proliferation and biomass incorporation. Physicians have long exploited this effect about cancer. We have been able to use it to image tumor in the human body by using the fluorodeoxyglucose FDG by positron emission tomography. This is all about the metabolism, altered metabolism, which is